Thank you for coming along to listen to me. Thank you to the organisers for the invitation to present at this excellent congress. I've had a great time. I particularly liked lying on the beach this morning. It was very warm and sunny compared to Edmonton, Alberta. So I'm not Canadian, I'm from the UK, but I've been in Edmonton for some 6 years. I'm going to talk about some preclinical work, and I know many of you are psychiatrists, and what happens at the preclinical level tends to stay at the preclinical level for several years, but some of what I'm talking about may get into the clinic at some point. But I'm going to talk about imidazoline ligands, and I'm going to try and convince you that there are targets for imidazoline ligands such as depression, pain and schizophrenia. I'm going to keep the talk short, so I'm not going to talk about imidazoline binding proteins, but I will talk about one of the endogenous ligands as a potential therapeutic target. I'm particularly going to focus on this endogenous ligand agmatine, which Fazer in the audience has worked on. And I'm going to try and persuade you that it may be useful to treat ethanol withdrawal, opiate withdrawal, depression, pain, and schizophrenia. I'm assuming that many of you won't know what imidazoline receptors are. Many years ago, it was noted that imidazoline ligands that bound to alpha-2 adrenoceptors also had effects through imidazoline receptors, and over the years these have been separated into I1, I2 and I3 imidazoline receptors, and they have this imidazoline moiety on all the drugs. And the proposed endogenous ligand for these receptors is Harman and agmatine. These are the typical ligands for the three distinct receptors. So I1 receptors bind moxonidine, and moxonidine's an antihypertensive, and it's these receptors that are said to control blood pressure in mammals and man. I2 receptors bind 2-benzophenyl imidazoline, and this controls monoamine turnover in the brain. And I3 receptors are found in the pancreas and control insulin release. So there are three distinct receptors. Now, why have they any interest in psychiatry? Well, this was a table put together by Garcia Sevilla in Spain, and it shows how imidazoline 2 receptor density changes in human brain in a number of conditions. For example, aging. As the brain ages, imidazoline receptor density increases. He has done experiments on brains taken from suicide completers and people with depression, and he's seen a large drop in imidazoline receptor density, so some 40%. In opioid-addicted brains or people, there is a drop in density of imidazoline receptors. Interestingly, no change in alcohol abusers. Work from my lab showed that in Parkinson's disease there is no change in the density of imidazoline receptors, but we also looked at Huntington's diseased brains and saw over 50% drop in the density of imidazoline receptors. And also glial tumours have a huge density of imidazoline 2 receptors. So in a range of conditions in the human brain, these receptors change for some reason, whatever reason we're not entirely clear on. But this has prompted the development of PET ligands to image imidazoline 2 receptors, and more or less the first successful PET ligand was published this year by a Japanese group, and they're doing animal studies at the moment. So this is in rat brain. They've got a fluorinated, or rather a C11 PET ligand. They've been injected into rats to image these imidazoline receptors throughout the rat. So hopefully this ligand will go into the clinic to image various conditions in the human brain, in particular gliomas. So what is an I2 binding protein? Well, it's been identified as a common motif on a number of proteins now. We know there's a binding site on monoamine oxidase, and we know some of these ligands inhibit monoamine oxidase. My group's focused on a binding domain or a motif on creatine kinase, and creatine kinase in the brain regulates energy. My group's also identified a I2 binding domain on NSEC1 binding protein, which is involved in vesicular fusion. So there's subtypes of I2 receptors. So I'm going to show you some evidence that they have antidepressant activity, they may be useful to treat pain, and some recent ligands that we've looked at are dopamine D2 agonists or partial dopamine D2 agonists that may be useful to treat schizophrenia. So there's some of the ligands that we've used. A couple of them I'm going to talk about. 2BFI, a typical I2 ligand. BU224, BU stands for Bristol University in the UK, and there's a couple of other ligands here. But you'll see they're fairly similar. They all have this imidazoline moiety on their structure. Some of the work in my lab focuses on rat brain microdialysis. I talked about this the other day, but we can place a dialysis probe into the rat brain under anaesthesia, and the rat recovers, and we can monitor monoamine turnover in the rat brain. And just briefly, this is two of our ligands, 2BFI and BU224, and we can look at extracellular noradrenaline versus time, so we can measure the levels of these monoamines. And here you'll see 2BFI dose-dependently elevates noradrenaline in the rat brain, and this is rat frontal cortex. And again, BU224 does exactly the same thing. It elevates noradrenaline in rat frontal cortex, and this may or may not reflect an inhibition of monoamine oxidase. So we know these ligands regulate monoamine turnover. So again, work from my lab, we've looked at a couple of ligands now, but I'll show you this one. This is the pour salt swim test, where we force rats to swim in a beaker of water, and normally rats will despair, they will give up, they will stop swimming, and typical antidepressants increase swim time in this test. And here you'll see a saline injected rat, and this is immobility in seconds. And here you see that BU224 decreases the time the rat stays stationary. So it increases the time the rat spends swimming, and this is indicative of this drug being antidepressant. And we've looked at a number of imidazoline 2 ligands in this paradigm, and the evidence suggests that they are potentially antidepressant in activity. Unfortunately, none of these compounds are what we call novel. They're not patentable, and if they're not novel, then drug companies are not interested. We need industrial interest, and we need novel compounds that can be patented. So what about a potential treatment for pain? I'll present some evidence for the use of these compounds to treat pain. I'm not going to go into the model in great detail, but when you place a rat's tail in warm water, if it's fairly hot, the rat will withdraw its tail after a period of time. And if you give drugs such as morphine, the rat leaves its tail in the warm water. And here you can see that morphine relieves the pain induced by the hot water. So the rat will leave its tail in the hot water. And again, here's another drug, tramadol, a good pain reliever. And in this paradigm, the imidazoline ligands 2BFI and BU224 do not give any pain relief to the rats. However, in the same study by Thorne et al., you see here, this is the effect of morphine in the absence of any drug, and you'll see that agmatine, the endogenous ligand, shifts this curve to the left. So with agmatine, you need a lower dose of morphine to get the same level of pain relief. Here's again the same thing for 2BFI. So adding 2-BFI to morphine, you get a better effect of relief of pain when you combine the two drugs. The strange thing about this study was that BU224, again, BU224 should act like 2-BFI. It didn't actually do anything. In fact, BU224 blocked the effect of 2-BFI and blocked the effect of agmatine. And I won't go into the bottom, but these data show the same for tramadol, that agmatine enhances the pain relief caused by tramadol. So you can use a lower dose of tramadol to get the same level of pain relief. This is another study by Lee et al. In 2011, this year. Again, in this study, you have morphine giving a degree of antinociception or pain relief. And he found, or this group found, that 2-BFI and BU224 alone were antinociceptive. So they gave a degree of pain relief, whereas agmatine did not. But again, that group also found that agmatine would enhance the antinociception or antinociceptive effect of morphine. So clearly there's something going on between these two groups of drugs. So I'm going through this briefly because I want to go on to some other work by Fazer. Finally, there's been much interest in partial D2 dopamine antagonists, or agonists rather, and their use in schizophrenia. So aripiprazole has been found very useful to treat schizophrenia. So in combination or in collaboration with a group in Italy, we've been looking at some new analogues of 2-BFI. So we've looked at some derivatives of 2-BFI as partial or full agonists at the dopamine D2 receptor, and we published this last year. So it's in collaboration with a group in Camerino in Italy. And again, I'm not going to go on into too much detail, except to say that we found 2-BFI was a partial D2 agonist, and we've now developed derivatives that are either full agonists, equivalent to dopamine, or partial D2 agonists. And in behavioral studies, these compounds increase locomotion and rearing, which would be indicative of dopamine activity. Unfortunately, we need industrial sponsorship to look at these drugs in models of schizophrenia and Parkinson's disease. So at the moment we're looking for further funding to look at these new novel compounds. So we need funding for further work, but I would add that given that aripiprazole we know works in the clinic, then the proof of concept has probably been and gone, so I'm not too hopeful of further funding. So perhaps more exciting is looking at work on the endogenous ligand, agmatine. Agmatine's been known as the endogenous ligand for over 10 years now. It's a very simple molecule that's been found in the brain and potentially has many therapeutic uses. And much of this work has been done by Fazer, who is thankfully here in the audience, and I'll show some of her experimental work, but also work by another Turkish professor. I don't know whether he's here. Is he? No. Well, this is one of his reviews that's in press at the moment. And you'll see that agmatine is derived from L-arginine and is then broken down by agmatinase. And you'll see normally it's in terminals, it's released from terminals and can act at alpha-2 adrenoceptors. It acts at these imidazoline receptors, and then more interestingly, agmatine can modulate the activity of N-methyl-D-aspartate receptors, which has been talked about at this meeting yesterday. So agmatine is a very interesting molecule. And here is some work from some 5 years ago showing how agmatine attenuates ethanol withdrawal. So these were rats that were addicted to ethanol, and you'll see various behaviors, and I can't read them from here, but stereotypy, tremor, wet dog shakes, and agitation. So when rats are withdrawn from ethanol and given saline injections, they get very agitated, they get wet dog shakes. But if you give these rats doses of agmatine from 20 up to 100 milligrams per kilogram, you can see how that attenuates the alcohol withdrawal. So agmatine's blocking that withdrawal syndrome. Some of Phaser's work. You can get rats addicted to morphine by putting a morphine pellet under the skin, and then after a few days you can precipitate withdrawal using naloxone, and this is one of Phaser's models. And here again there's various behaviours that we look at, wet dog shakes, etc., and they're all indicative of a severe withdrawal syndrome. And you'll see quite clearly that in control saline treated rats, they get very agitated, you get lots of wet dog shakes, tremors, etc. But agmatine suppresses the opioid withdrawal syndrome. So in some way agmatine can block opiate withdrawal. Further work by Phaser again. These are sham rats now, so agmatine in sham rats doesn't do anything to their behaviour, so there's a good control. But here we have morphine addicted rats, and when they're withdrawing we see jumping, defecation, tremor, teeth chattering, and all of these are blocked by agmatine. So agmatine will block naloxone precipitated withdrawal in this rat model. Again, more interesting work from Phaser's group has shown that agmatine is active in the pour salt for swim test. So here we have saline injected rats, and here we have a control drug, imipramine, a tricyclic antidepressant, so reduced immobility. And here we have doses of agmatine doing exactly the same thing as imipramine. So again, agmatine is active in the pour salt swim test, indicative of antidepressant activity. And here is some data showing the effect in an open field paradigm, where the rats prefer to stay in a closed field rather than open field. And imipramine, rats will go out into the open arms of a maze, and agmatine at certain doses will mimic this effect, indicating that agmatine may have a use to treat anxiety. Pain. I can't remember which pain model this was using, but it doesn't matter. Here we have latency again, and you'll see that rats injected with saline feel pain, so they'll withdraw their limb or tail. But over time, rats given different doses of agmatine will have an increased latency. They don't feel the pain. So again, indicative that some way agmatine is attenuating the pain. I mentioned earlier that agmatine is broken down by an enzyme, agmanitinase, and John Pilletz in the States has now issued a patent looking for compounds that will block agmanitase, so therefore agmatine will be around longer and therefore will be able to prevent, and I've just highlighted, or would be useful to treat opioid addiction, idiopathic pain and depression. So if compounds that have blocked the enzyme may be useful to treat these conditions. So certainly John Pilletz is interested in this idea. Now in a paper that's not out till next year, someone has been looking at the enzyme agmanitinase in hippocampus of patients, and he's found that the actual enzyme is upregulated in the hippocampus of the brains of deceased patients. So clearly this enzyme is doing something that's critical and is involved in mood. And again, this paper comes out in 2012. Finally, there's a paper also coming out in 2012 that indicates that agmatine may be useful to treat schizophrenia. Again, I won't go into detail about this test, but this is what we call a conditioned avoidance test. And here we have percentage inhibition of conditioned avoidance, and this is a saline control. And here we have agmatine at different doses causing inhibition of conditioned avoidance. Here we also have haloperidol having a minor inhibition, but what's interesting about this study is when they combine the very low dose of haloperidol with a low dose of agmatine, we get this pronounced effect. So a low dose of agmatine that doesn't do much at all, 20 mg per kilogram, enhances the effect of haloperidol. And the same is true for olanzapine. So agmatine is enhancing, or potentially enhancing, the anti-schizophrenic effects of these drugs. In the same study, they looked at the effect of agmatine on amphetamine and ketamine-induced behaviors. And again, this is a kind of model for drugs to treat schizophrenia. And here you see the saline controls, and here we have amphetamine-treated animals where they get hyperactive, and then agmatine blocks that hyperactivity. Ketamine also induces hyperactivity, and agmatine will block that hyperactivity, again suggesting agmatine combined with possibly another drug may be useful to treat schizophrenia. So that's some simple data from the preclinical level suggesting that based on animal studies, agmatine analogues may be useful to treat ethanol withdrawal, morphine withdrawal, depression, anxiety, pain, and schizophrenia. So before I finish, you'll see that agmatine is not stable. It's broken down. Therefore, we have been looking for and collaborating with chemists in Poland to look for stable agmatine analogues. And this year we've published on stable agmatine analogues with one of my students, and I'll show you the structures here. Here we have agmatine. The chemist had the idea to fuse in this molecule here, to fuse in this imidazoline molecule here to give us stable agmatine analogues. And so we've now been looking at these stable agmatine analogues. You can build on the side of these structures here to get compounds that retain affinity for the imidazoline binding sites. We hope that they will cross the blood-brain barrier and in animal models will be shown to be active. So we now require further testing of these stable agmatine analogues in models of addiction and models of pain, etc. So if anyone's got some spare euros they don't want, we would welcome some investment to look at these, I consider potentially exciting stable derivatives of agmatine that we hope will be used to treat ethanol withdrawal or opiate withdrawal, depression, anxiety, etc. So fingers crossed. As I'm sure some of you may know, you can pick up agmatine in health food shops now. It's widely used and abused, I suppose, for people who wish to keep fit. But I don't know whether they look at their mood at all. So, thank you for listening. This work's funded by the Natural Sciences and Engineering Research Council of Canada. And thank you for listening.